The enhanced biological phosphorus removal process can be implemented in activated sludge systems by recycling the mixed liquor activated sludge through alternating anaerobic, aerobic, or anoxic stages, while directing the influent, usually rich in volatile fatty acids like acetate and propionate, to the anaerobic stage. This promotes the enrichment of the system with polyphosphate accumulating organisms. PAOs can store up to 0.35 to 0.38 GP/GVSS. In contrast, ordinary heterophobic organisms can only store around 0.3 GP/GVSS, approximately 10 times less phosphorus. PAOs can be enriched under these alternating conditions because they have the capability to use intracellular polyphosphate to produce the required energy in the form of adenosine triphosphate, to store VFA as intracellular polybeta hydroxyalkanoates. Stored PHAs are, later on, utilized under aerobic or anoxic conditions for enhanced phosphorus uptake, glycogen synthesis, biomass growth, and maintenance. Actual phosphorus removal can be achieved by wasting the mixed liquor after the aerobic phase when PAOs have their poly-P pools full. As such, in the liquid phase during the anaerobic stage, the orthophosphate concentrations increase due to poly-P hydrolysis, whereas the VFA concentrations decrease because of the VFA storage as PHA. In the PAO biomass, the PHA content increases while the poly-P and glycogen contents decrease. In the aerobic or anoxic stages, when oxygen or nitrate is present, orthophosphate is removed from the liquid phase to replenish the intracellular poly-P pools. Within the POA biomass, the PHA content decreases while poly-P and glycogen storage pools increase. Theoretically, no major biological processes occur in the settling stage, which can differ in practice. There are different anaerobic parameters of interest to assess the metabolism and physiology of the EBPR process. For instance, the maximum specific acetate uptake rate, RAC, AN, provides information about the consumption and storage of VFA. The maximum specific P release rate, about the poly-P hydrolysis process, and the endogenous P release rate, about the maintenance process of the cell. To study the stoichiometry, it is important to determine the total acetate consumed, as well as the net P release caused by the acetate uptake process, after excluding the P release caused by the endogenous P release. Among the aerobic and anoxic parameters, the maximum specific aerobic P uptake rate and the maximum specific anoxic P uptake rate are two of the most important parameters of interest. Nevertheless, other rates are rather useful to assess the PAO metabolism depending on the nature and purpose of the study. To illustrate the execution of EBPR batch activity test, this lecture will show the conduction of an EBPR.AER.4 test, which has the main purpose to assess simultaneously the aerobic and anoxic EBPR capacities of a given sludge. This test starts with the anaerobic incubation of sludge in Reactor 1. All carbon source should be taken up within the anaerobic stage. Thus, the reaction time should be long enough or the carbon concentration adjusted to secure full uptake of carbon. In this example, the length of the anaerobic incubation will be 2 hours and 400 micrograms COD/L will be added to an enriched EBPR sludge. After the anaerobic stage, usually 50% of the sludge volume is transferred to a second reactor where the aerobic stage will take place. Thereafter, nitrate is added to Reactor 1 to create anoxic conditions, while air is supplied to Reactor 2 to generate the required aerobic conditions. Usually, the duration of the anoxic and aerobic stages is similar and at least as long as or longer than the anaerobic incubation. Throughout the conduction of the test, several samples for particulate and soluble compounds need to be collected to assess the anoxic and aerobic conversions. The planning, preparation, and execution steps, operating conditions, and sampling times can be organized in a working sheet. The working sheet can be used prior, during, and even after the test for archiving and reference purposes. The working sheet needs to include the sampling campaign, detailing the sampling times and the compounds of interest for the anaerobic and anoxic stages. As well as the sampling program for the aerobic phase and for the solids concentrations in the start and end of each stage, the working sheet can include a table to present a summary of the concentrations of solids and intracellular compounds measured in the different stages. This is useful to estimate and present the active biomass fractions. This test is performed in two similar batch reactors. In Reactor 1, the anaerobic aerobic test is carried out, and in Reactor 2, the aerobic test. Both reactors are similar, but Reactor 2 is smaller, because half of the volume of Reactor 1 will be transferred to Reactor 2 to conduct the aerobic test. Prior to the execution of the test, verify the correct operation of the biocontroller and sensors. They need to be in good operating conditions and well calibrated. Also, confirm that the whole setup is ready to carry out the activity tests. Prepare your working sheet in accordance to the execution of the test and keep it close to the setup together with a stopwatch. They will be the main guide to conduct the test. Place all your working material next to your setup and confirm that all required solutions, materials and consumables are ready. The sampling cups and tubes need to be clearly labelled and organised to facilitate the collection and preparation of the samples during the test. Among other materials and consumables, verify that there are sufficient filters, syringes and paper detection strips. Do not forget to have the required safety gloves and other personal safety equipment and materials. After the sludge has been collected and, if applicable, washed, it should be carefully mixed. Mix gently to obtain an homogeneous sample, avoiding that the biomass settles and remains at the bottom of the bucket. Once the sludge has been well mixed, pour it carefully into the reactor. Once the sample has been poured into the reactor, remove all the materials used to pour the sludge sample and close the reactor. Then start the mixing. Verify that the sludge sample is well mixed and that there is neither sludge stratification nor dead volumes. Otherwise, gradually increase the mixing speed until the sludge is well mixed. On the opposite, if the mixing speed is too fast and even violent, then reduce it gradually. Verify that the parameters of interest, like pH and temperature, are within the recommendable operating ranges. If not, correct and wait until they are stable and within the required allowable intervals. A preliminary sample can be collected to know the initial characteristics of the sludge. In particular, nitrate should be absent because it can interfere with the creation of anaerobic conditions. To avoid the intrusion of oxygen, connect the syringe tightly. Push and pull at least 5 times. Avoid the accumulation of bubbles in the syringe by tapping the syringe until the air bubbles are released from the syringe. Carefully remove the syringe. Use a lab clip to close the sampling port and avoid spillages. Filter the sample through filters with a pore size of 0.45 micrometers. Tightly connect the filter to the syringe. Avoid pushing too strongly or the filter may break and your sample will be contaminated. Use a fast nitrate estimation method, like paper detection strips, to quickly assess the presence of nitrate. If nitrate is detected, slow down the mixing speed and wait for at least 30 minutes whilst carefully and continuously monitoring the system. After the waiting time, repeat the same procedure for the detection of nitrate. Take another homogeneous sample. Filter the sample and assess the presence of nitrate. If nitrate is no longer detected, verify that the sludge and the operating parameters are stable and adjust the mixing speed to the one recommended for the conduction of the test. If after 30 minutes no nitrate is present in the sludge sample, add allyl N-thiourea. ATU will inhibit nitrification when the sludge sample is aerated. Pour the ATU stock solution in a beaker and take the corresponding volume with a syringe. This will avoid contamination to the stock solution. Usually, 20 milligrams per litre of ATU are sufficient to inhibit nitrification. Calculate the ATU volume to be added based on the concentration of the stock solution and the expected dilution rate considering the reactor volume to reach 20 milligrams per litre of ATU inside the reactor after dilution. Add the ATU volume with the syringe through an opening on the lid of the reactor. Check that the sludge is well mixed, otherwise increase the mixing speed. Thereafter, start up the aeration. Leave the sludge under aerobic conditions for at least 30 minutes. This will help to remove any biodegradable organics present in the sludge sample and avoid that they could interfere with the anaerobic phase of the batch activity test. Regularly confirm that the concentration of dissolved oxygen is higher than 50% of the saturation concentration and that the pH is stable at the desired set point. Confirm the well mixing conditions of the sludge and that the dissolved oxygen concentration and pH level are within the desired operating ranges. If everything is in order, stop the aeration to take the first samples from the reactor. This will reduce the presence of air bubbles in the samples. Reducing the error and deviations in the samples collected for the determination of suspended solids, PHA and glycogen. Tightly connect the syringe. Pull and push at least 5 times to take a representative and homogeneous sample from the reactor. Tap gently the syringe to remove any air bubbles. Take the required volume for the corresponding analysis. Carefully close the sampling port and remove the syringe. Usually, between 10 and 12 millilitres are collected for the determination of PHA and glycogen contents. Commonly, the samples are poured into plastic centrifuge tubes. The tubes contain 2 to 3 drops of formaldehyde to preserve the sample. Quickly open the tube and pour the sample. Close the tube and invert the tube upside down at least 3 times to mix the sample with the formaldehyde solution. Thereafter, place the sample in ice whilst the rest of the samples are collected. For the determination of PHA and glycogen contents, usually 3 samples are collected in the same manner. Place the second sample in ice. Continue with the collection of the third sample for the determination of PHA and glycogen. After the collection of the third sample, continue with the collection of the samples for the determination of the solid's concentration. Follow the same sampling procedure as for the previous samples. Usually, the samples are taken by triplicate due to the relatively high error of the analytical techniques. The volume of the samples for the determination of the concentration of solids can be poured in plastic cups. The plastic cups with the samples of solids should also be stored in ice, whilst the rest of the samples are collected. Place the second sample in ice and proceed to take the last sample for the determination of the concentration of solids. Place the last sample in ice. Once the samples for particulate compounds are taken, continue to take a filtered sample for the analysis of soluble parameters. This is also the last preliminary sample of the test. Apply the same sampling procedure. Take the required volume for the analysis of the parameters of interest, which usually comprise soluble COD, acetate, orthophosphate, and ammonium. Filter the sample through filters with a pore size of 0.45 micrometers. Avoid applying excessive pressure or the filter may break. The sample will get contaminated and it would need to be taken again. If the filter gets clogged, replace it and continue with the filtration procedure. Prepare and have ready at least 2 or 3 filters per sample. Once all samples have been collected, place them temporarily in ice prior to their proper storage and analysis. Check the corresponding steps in the working sheet and mark or cross out the steps that have been completed, as well as the samples that have been collected. Verify the stability of the sludge by assessing the pH, dissolved oxygen concentration and temperature. If stable, start the supply of nitrogen gas for 5 to 10 minutes. This will remove the dissolved oxygen present and create anaerobic conditions. The dissolved oxygen concentration should drop below detection limits to consider that strict anaerobic conditions have been reached. Continuously verify the stability of the parameters of interest, like dissolved oxygen concentration, pH, temperature, and mixing. Verify the next steps in the working sheet. Confirm visually the stable operation of the reactor. Also confirm the stability of the parameters of interest. In particular, that the dissolved oxygen concentration has dropped below detection limits, indicating the creation of anaerobic conditions. Prepare the addition of the carbon source of interest. In this example, acetate will be added as carbon source. Pour the acetate stock solution into a beaker. Using a syringe, take the volume of acetate to be added. This volume needs to be carefully determined considering the target initial concentration after the acetate is diluted inside the reactor, the concentration of the stock solution, and the expected dilution rate based on the reactor volume. Collect the volume, avoiding the presence of air bubbles. Open the reactor and add the carbon solution quickly. Simultaneously, start up the stopwatch and close the reactor. This will be the actual beginning of the test. Confirm that all operating parameters are within the allowable operation ranges. Verify the past, current and future execution steps and mark the completion of the addition of substrate in the working sheet. After the addition of the carbon source, confirm the stability of the system. Follow the sampling program and start to take the first sample, at least 20 or 30 seconds before the sampling time, indicated in the working sheet. This time interval is needed since it can take several seconds to collect and filter the sample. This will help to determine the kinetic rates with precision. Follow the sample procedure previously applied for filtered samples. This example only illustrates the collection of filtered samples. If samples for the determination of solids or particulate compounds need to be also collected, then a longer time interval is needed to collect and prepare all the samples in time. Place the sample in ice after collection and filtration. Mark the completion of the sampling step in the working sheet. Thereafter, continue with the sampling program as planned and described in the working sheet. Once the execution of the anaerobic stage has been successfully conducted, confirm the following steps needed to complete the anaerobic stage. Verify that the reactor continues under stable operating conditions and that all parameters are stable. Stop the supply of nitrogen gas to collect the final samples for the determination of particulate compounds, as planned in the working sheet. Follow the same sampling procedure previously presented. Samples need to be collected in the beginning and end of the anaerobic stage for the determination of intracellular compounds, like PHA and glycogen, to determine their anaerobic conversions and the anaerobic stoichiometric ratios of the process. Remember to invert the sampling tubes upside down a couple of times to mix the sample with the formaldehyde solution and favour the preservation of the samples. Place all samples in ice temporarily while they are stored and analysed. Take the samples for PHA and glycogen determination by triplicate. Also, collect the samples to measure the solids concentration by triplicate, since they are needed to determine the PHA and glycogen contents given that the PHA and glycogen analysis are usually provided as percentages of the dry solids concentrations. Collect the last filtered sample of the anaerobic stage to determine the final concentrations of the soluble compounds of interest, like soluble COD, acetate, orthophosphate, and ammonium, as planned in the working sheet. Remember to prepare enough filters and filter the sample carefully. The concentrations measured at the end of the anaerobic phase will correspond to the initial concentrations of the subsequent anoxic and aerobic stages. It is important to underline that all the samples need to be collected before the end of the anaerobic phase. Thus, allocate enough time for sampling before the end of the anaerobic stage. Confirm that all required samples have been collected before concluding the anaerobic stage. After the anaerobic stage, restart the supply of nitrogen gas to reactor 1 and also supply nitrogen gas to reactor 2 to create anaerobic conditions. Thereafter, transfer the sludge from reactor 1 to reactor 2. During the sludge transfer, do not turn on the stirrer. Avoid mixing the sludge and keep the supply of nitrogen gas. This will minimise the intrusion of oxygen and will also help to maintain the sludge under anaerobic conditions. Follow up how Reactor 2 is filled and stop the sludge transfer when the desired volume in Reactor 2 is reached. Once the sludge has been transferred, turn on the stirrer in Reactor 2. Confirm that the conditions remain stable. Prepare the nitrate solution to create the anoxic conditions in Reactor 2. Pour the stock solution into a beaker and take the required volume with a syringe. Add the required volume of stock solution. Consider the concentration of the stock solution, the estimated water volume left in Reactor 1 after the sludge transfer, and the target initial nitrate concentration in the beginning of the anoxic stage. The anoxic phase will start when the nitrate solution is added. To carry out simultaneously the anoxic phase in Reactor 1 and the aerobic phase in Reactor 2, start up the air supply to Reactor 2 2.5 minutes after the nitrate solution is added to Reactor 1. This will create a delay of 2.5 minutes in Reactor 2 with regard to Reactor 1 and facilitate the simultaneous sampling of both reactors. Confirm that the operating conditions are stable and get ready to start the simultaneous sampling of both reactors. Confirm the stability of the parameters of interest at the corresponding operational set points. Keep track of the time to start the collection of samples. Following previous recommendations and the sampling plan, first, collect the sample of the anoxic phase in Reactor 1 and thereafter the sample of the aerobic phase in Reactor 2. To have enough time for sampling, start at least 20 to 30 seconds before the target time indicated in the working sheet. Mostly because for a precise determination of the kinetic rates, the filtration process needs to be done at the time indicated in the working sheet. The delay of 2.5 minutes will facilitate the sequential collection of samples in reactors 1 and 2. In this example, only the collection of filtered samples will be shown. The collection of samples for the determination of particulate compounds like PHA and glycogen can also be carried out. But the collection of samples for particulate compounds will imply to allocate more time for their collection, since also these samples need to be collected by triplicate. Thus, take the first sample of the anoxic stage created in Reactor 1 and place it in ice. Then, 2.5 minutes after the collection of the first sample in Reactor 1, continue with the collection of the first sample in Reactor 2, which corresponds to the first sample of the aerobic stage. As previously recommended, collect, filter, and place the sample in ice temporarily until it is properly stored, preserved, and analysed. Confirm that the required samples have been collected and mark it in the working sheet. Thereafter, continue with the simultaneous sampling of the anoxic phase in Reactor 1 and of the aerobic phase in Reactor 2 as planned and indicated in the working sheet. Keep adequate track of the execution and simultaneous sampling of both reactors. Verify and confirm that the operating parameters are stable. By the end of the anoxic stage in Reactor 1, collect by triplicate the corresponding samples for the determination of PHA and glycogen contents. Follow previous recommendations for the collection of these samples. Their collection is essential to determine the conversions of intracellular compounds in the anoxic stage executed in Reactor 1, and thereby to determine the stoichiometric ratios of the anoxic conversions. Once the 3 samples for the determination of PHA and glycogen have been collected and placed in ice, also take by triplicate the required samples for the determination of the solid's concentration. Place the samples collected for the determination of the solid's concentration in ice. Take the last filtered sample for the determination of the soluble compounds. Place and keep all samples in ice. Confirm that all samples from the anoxic zone were collected and mark it in the working sheet. Once the anoxic zone has been completed in Reactor 1, confirm the latest status of the aerobic phase in Reactor 2 and collect the final samples of the aerobic phase. Verify that all parameters are stable. stop the supply of air to reactor 2. This will reduce the presence of air bubbles in the samples. In particular, in the samples collected for the determination of particulate compounds like PHA and glycogen, or solids, proceed to collect the samples for the determination of the intracellular compounds. Follow previous recommendations, like regularly tapping the syringe when collecting the sample to reduce the presence of air bubbles, to take a representative sample by pulling the plunger back and forth, at least 4 to 5 times. Collect the samples by triplicate and place them temporarily in ice. Take the required samples for the determination of the concentration of solids by triplicate, and also place the samples temporarily in ice. These last samples for the determination of intracellular compounds and concentration of solids will be useful to determine the total conversions of these compounds in the aerobic phase. Meanwhile, the filtered samples will be useful to determine the conversions of the soluble parameters, in particular of orthophosphate. Remember to carefully filter the samples, avoiding too much pressure and having enough filters to collect the required filtered volume. When all samples have been collected, confirm that all steps and activities have been completed and mark it on the working sheet. After the execution of the test, the same working sheet used for the test execution can be filled out, with the analytical data determined for the different samples. Thus, the results of the analytical analysis from each sample can be added to the working sheet. Also, the results from the samples collected in the anoxic and aerobic stages can be added. The results of the determination of the concentrations of solids measured in the beginning and end of each stage can also be included. At this stage, the summary table located at the bottom of the working sheet gains particular importance, mostly because it can be used to display and calculate the corresponding intracellular compounds at the beginning and end of each phase, and also the active biomass fractions. In this example, the MLSS, MLVSS, the MLVSS stroke MLSS ratios, and the ash content can be added. These concentrations and their ratios can be calculated based on the average values obtained in the table shown previously. They are computed for the start and end of the anaerobic stage, which corresponds to the beginning of the anoxic and aerobic stages. Also, the concentrations at the end of these stages can be added. Thereafter, the pHb and pHv concentrations measured in the start or end of each phase can be added and their sum reported as the total pHa concentration. If other pHa compounds are measured, like pH2MV, an extra line should be inserted and the concentration of any additional pHa compound needs to be added to the total pHa concentration. The glycogen concentrations measured at the start or end of each phase should also be included. The percentage of pHa plus glycogen per ml/VSS concentrations can provide an estimation of the volatile suspended solids composed by these intracellular compounds. Thus, the active biomass concentrations can be computed by subtracting the pHa and glycogen concentrations from the ml/VSS concentration. If desired, the active biomass can be expressed in terms of cmmol units by dividing the concentration originally expressed in milligrams per litre by the conversion factor 26 milligrams active biomass per cmmol. Since the fractions of intracellular compounds vary along the EBPR tests, affecting the ml/VSS concentrations, the active biomass is a useful estimation of the actual concentration of microorganisms that play a role in the EBPR conversion processes. The data of the analysis from each sample can be plotted as a function of the sampling time. The time when substrate was added should be taken as the starting point. Overall, two stages or phases can be identified: the anaerobic stage from 0 to 2 hours, and the anoxic and aerobic stages from 2 to 4 hours. However, since the samples of the anoxic and aerobic stages are collected simultaneously, they overlap between 2 and 4 hours. To facilitate the visualization of the data, similar markers can be used for similar compounds. For example, circles for glycogen, diamonds for PHA, or triangles for PO4P, but using different colours, black and white or greyish levels, to identify whether they correspond to the anaerobic in black, anoxic in grey, or aerobic stage in white. The same graphic can be used to estimate the volumetric conversions of the compounds of interest. For instance, for the anaerobic uptake of acetate and the orthophosphate profiles in the different anaerobic, anoxic, and aerobic stages, the maximum volumetric conversion rates can be estimated by linear regression. For the linear regression, only the first samples measured at the beginning of each stage should be taken into consideration. Ideally, more than 5 data points should be used, while aiming to reach the highest determination possible. The maximum volumetric conversion rates, in this example, in milligrams per litre per hour, will correspond to the slope of the linear regression expression. As observed, a negative slope indicates that a compound is consumed, while a positive slope means that the compound is produced or released. The anaerobic conversions can be determined based on the differences between the initial and final concentrations of acetate, orthophosphate, PHA, and glycogen, measured at the start and end of the anaerobic stage. Similar to the slopes of the kinetics, positive values mean that the compounds have been produced like PO4P and PHA, whilst negative values mean that they have been consumed like acetate and glycogen. Using the anaerobic conversion values, stoichiometric ratios such as the PO4P to HAC, PHA to HAC, and glycogen to HAC can be computed by dividing the conversions of PO4, PHA, and glycogen by the HAC conversion value. Once they have been computed, these values can be compared to those displayed in Table 2.2.3 from the textbook. This is useful to assess whether the stoichiometric values are representative or not of EBPR cultures. In this example, the ratios of 0.53 pmol by cmol, 1.42 cmol PHA by cmol HAC, and 0.41 cmol glycogen by cmol HAC are similar to those reported by Smulders et al. In 1994 for PAL cultures enriched with acetate as carbon source. Also, these ratios are comparable to those from DPAO cultures enriched in lab systems with acetate as described by Kuba et al. In 1996. This indicates that the system is highly enriched with PAO or DPAO. If the ratios differed considerably from those displayed in Table 2.2.3, the data needs to be carefully assessed and compared to other values displayed in the same table. If the values differ considerably, the potential influence of certain factors should be considered, such as pH, temperature, or carbon source, and even the presence of other microorganisms, as discussed in sections 2.2.6 and 2.2.8 from the textbook. The slopes of the linear regressions of the different profiles correspond to the maximum volumetric conversion rates. In this example, the rates are expressed in milligrams per litre per hour units. When these rates are divided by the active biomass concentration, then the maximum specific rates can be determined. In the upper part of this table, the rates are expressed in C-mol or P-mol units, and in the lower part of the same table, in milligrams per litre units. As required, the measurements can be originally presented in either units if they are converted using the unit conversion coefficients, presented in Annex 1 in the textbook. Similar to the anaerobic stoichiometric ratios, the kinetic rates can also be compared to those previously reported in literature, like those presented in the textbook. For the anaerobic kinetic rates, the values presented in Table 2.2.4 can be used for comparison purposes. As observed, the maximum anaerobic HAC uptake rate of 0.67 C-mol per C-mol per hour or 7.9 mgHAC per GVSS per hour is lower than the values reported for lab-enriched cultures, but higher than the rates observed in full-scale systems. These differences suggest that either the system is not highly enriched with PAO, or with other organisms capable of taking up acetate under anaerobic conditions. But also it cannot be discarded that they are not that active under the operating and environmental conditions created in the batch activity test. The maximum specific aerobic phosphorus uptake rate, with a value of 0.040 p-mol per c-mol per hour, or 48.1 milligrams PO4P per GVSS per hour, is in the range of previous values reported by Burjanovic and colleagues in 1997 of 0.046 p-mol per c-mol per hour, as presented in Table 2.2.6 of the textbook. But the Anoxic P uptake rate of 0.020 p-mol by c-mol per hour, or 24.1 milligrams PO4P by GVSS per hour, is higher than other values reported, as displayed in Table 2.27. In this example, the Anoxic P uptake rate is only half of the aerobic P uptake rate, 2.4.1 milligrams PO4P by GVSS per hour, compared to 48.1 milligrams PO4P by GVSS per hour, respectively. This suggests that about 50% of the PAO biomass present in the sludge is capable of performing the anoxic removal of orthophosphate, or that the total PAO biomass is composed by about 50% denitrifying PAO or DPAO. Whilst these values provide an estimation of the aerobic and anoxic activity of PAO and DPAO, the rates need to be carefully assessed, taking into consideration the potential effect or influence of the environmental and operating conditions that were used for the execution of the batch activity test, as discussed in section 2.2.8 of the textbook. Other operating, environmental and even microbiological factors may need to be assessed if the results differ considerably from the previous data reported in literature.